Hello, good morning, good evening. How are you? Hope you're well wherever you sit and listen. This is a medical cannabis show, which is all about the promotion of organic and safe practices and respecting the law. So if you're not into those things, you probably won't like this channel. Otherwise, for those that are into that sort of stuff, there should be some really good topics that'll be brought up. I'll show my university slides and try and get the points across, so understanding's a lot easier. All right, thank you. And today is an open topic. So there we go. There's the little slide thing for today. It's an open topic, so I hope you can bring any questions and start off and see where it ends up. Otherwise, I'll start showing some of my slides on whatever I want to show. And who knows, it mightn't be, you mightn't like it. It'll be plant and soil and microbe related, which is always steered towards medical cannabis. And yeah, try and get my point across that way with the slides. Nothing worse than sitting when I want to chat to you. You know, I'm without showing slides because you don't hear it. All right, so it's hello to Vinny or Vin. G'day, Vin. Hope you're doing well today. Please, if you've got any questions, throw them out. Lene, how's it going, Lene? Good vibes. Top of the morning to you. I have no volume. Uh-oh. No, I can see the little thing going. Hello. Yep, it should be working. I think it's just your volume. I hope. Is that right, Vin? It should be right, yeah, because this thing doesn't go up and down. Yes. Please turn your volume up, Lene. Righto. Where's, oh, yep, here we go. No, not remove. Add to stream. Yes. So that's today's. So it's just an open topic. So we'll see if there's going to be some things come up. Sunshine. G'day, Sunshine. How are you? Hello, hello. Vinny says, sounds fine. Thank you, Vinny. Very good. Sunshine's got a question. Is yogurt fermented milk good for soil? That's an interesting one. Is yogurt or fermented milk good for soil? So is lactobacillus species good for soil? Well, they tend to put out a very heavy biofilm and aren't really symbiotic. So to say that they'd be good for it, depends what you want to get out of your soil. But if you're looking for a full soil food web to be established, I don't think that would be good for that. Just because of that reason that they put out a heavy biofilm, and with that, that means that it's going to put all heaps of antibiotics and stuff in it, and that's going to kill other microbes and fungi. And so if they want to try and compete in that space, they won't be able to. So that's not good. Fermented. Wow. I'm just trying to think of some slides that I might have for that. I can show, what about some contaminants in the soil? No, that's not nothing to do with lactobacillus though. So I need the microbes. That might be to do with bioremediation. That's where it would have been under that. Oh, I can look through some of that. All right, let's look through some of that. Good question. I should say hello to the other people in the chat. I saw some like Dave, 9069, how are you? Ned Kelly, good morning, Ned Kelly. Buds and Hazards, nice to see you. So Sunshine's asked a question, is yogurt fermented milk good for soil? So I'm going to have a look through some slides that are going to be, I'll look through bioremediation. No, what's that course? It's hard to do, look and talk at the same time, but I try. So study experimental biotechnology. Is it that one? Environmental biotechnology. I think it's that one. Oh, a little more. All right, I'll pull it up. I'll show you some slides because it's to do with microbes. It's fun. And then I'll try and have to remember that question because I get off track with microbes because they're so, not entertaining, but they're so interesting. you can culture them so fast, you can study them. Well, only 99% of them has been able to see. Look at me already getting off the topic. 99.9%, I think. Actually, it depends where it is. That's in seawater. Haven't been cultured yet. Good morning, good evening to small tubes. There's a question up here, small tubes. Sunshine Rainbows has asked, is yogurt or fermented milk good for soil? So I'm going to try and show some slides in my environmental technology, biotechnology class, which was all about this one, microbial ecology. And I'm looking for lactobacillus relationship to soil. So if you see it, holler out, say stop, stop. But that's what I'll be looking for. Superbugs versus microbial reservation measurement. Engineered bacteria, activated sludge. Yes, that's so. Anyway, I have to think of the question. Lactobacillus. I'll get so sidetracked here because there's so many different interesting things. Retain slow-growing biomass. Yeah, look at the fast bone. Actually, I'm not going to get into it. More powerful analytical tools. Yeah, good. Anaerobic respiration. Death of microbes. What causes deaths of microbes? Is there a lactobacillus culture there? Nope, there's not. It's sulfur. A lot of sulfur ones. Properties, carbon dioxide, cell metabolism, function and growth. That's got nothing. See if you understand these basic principles, that's how you can manipulate cells, and then you can manipulate actions from those cells, and then you can get outcomes, favorable outcomes for experiments or things that you want to try and manipulate. They might call it genetic manipulation, but it's still how to do it. The following groups of microbes differ from relation to oxygen. This is the classifications. It's a big radox. Fungi. Types of fungi. No. Molds, yeasts, and mushrooms. You know, difference between molds and yeasts. Molds have no hyphae on them, and yeasts have hyphae. So, yeah, that's really about it. Algae, they're cool. Protozoa. Yes, how to make protozoa, how to do them, why they're beneficial. Helminths. Helminths are parasitic worms that survive in humans and animals. So elemental composition, culture mediums, no. Examples of culture media, no. Chemical contents of microbial biomass. Yeah, that's pretty cool. That's what's actually inside of the microbes itself. If you want to break down a microbe, you can see what is inside of it. Like it's got 70% water, 30% is all of this extra dry weight material, which comes over to here, which contains all these different things. But there's what's it called? Peptidoglobin is the cell wall. It's peptidoglycan, something glycan. Anyway, that's its breakdown over here through proteins into RNA, DNA, lipids. Oh, there it is, peptidoglycan. That's the cell wall constituent. That's what the cell walls of microbes are made of. And the soluble pool. So yeah, you can get stuff out of it. There's an NPK. Anyway, medium. I have to think about back to the topic of lactobacillus. True. There's the cell growth phases. I did a whole course on just sort of cell biology to understand this, so I could work with polyploids a lot better to know when is the right when is the right stage for the cell development to manipulate it. Prokaryotic cells. Yes. Growth yield. Yes. That's where it can grow 4,000 times around the planet if you get it in its right optimum growth. I'll explain that a bit. 4,000 times around the planet is the speed in which a microbe can be growth under its right conditions. So within 48 hours, it can go around the planet 4,000 times. That was that thing last week. I was talking about the different ranges of the compost, the mesophilic and the thermophilic ranges, and that's the temperature scale that governs those. Effects of oxygen, no. pH, Yeah, those things are all, like, remember for soil, it's three things. It's your microbes, your physical condition, and your biology. No, that's a biology. Temperature, physical and chemical. Biological, physical, and chemical properties make up the three things. Where here, there's 2, 4, 6 things that make up microbial activities. And at reproducible in tissue culture, there's 5 things the plant needs really for the cell. So you'll knock out the pH, because your pH will already be stable on what you make up your nutrient medium. Affects the nutrients. Yeah, a nutriophile. Isn't that cool? Just gobbles it all up. Carbon energy transfer. 2 important questions. What drives metabolism? And what are the energy and carbon sources? The answers provide fundamental nutritional basis for subsequent hypotheses and inquiries about past and ongoing biogeochemical and ecological processes. No. What's the lactobacillus? It's nothing to do with the chemolithotrophs. Synthetic sugars. Sorry, I don't think I'm going to have much luck here. Maybe it's in a different, well, it could have been a different class. The biogeochemical, ATP. Well, that's the cool, that's the cell wall. That's all its different things that it's transferring from outside the cell into the cell, from the protoplast inside. Types of reactions, mechanical. All right, I think I'm losing it. Fermentation. Look, there's a fermentation, but that's to do with the breakdown process, and that's when there's no oxygen involved. So it'll have to use oxygen as an electron acceptor in the form of nitrate or sulfate. So it's getting down to the anaerobic. So that's not really related to that question, but it sort of is because it's written there. I'm trying for you, sunshine. There it is. Like here, that's what I was just talking about then. So once the oxygen's gone, it'll start using this as time goes on, and if it's in a glass jar like that, this is how microbes work. So if the oxygen goes, it'll look as nitrate as the oxygen source and pull it out of that, and the denitrifiers will be happy, and then it'll look for the OH in the magnesium, and then the iron, it'll look for the same and pull it out of that, and sulfate, and then back when the methanogens in the methanogenic archaea. But you're going to use all the CO2, which is reduced from all of these microbes. They use that as their electron acceptor. This is supposed to be an electron accepting process. There you go. This is a similar thing that I was just explaining. That's how oil breaks down anyway. Your catabolism and animalism equal your metabolism. And your catabolism makes it the ATP, and your animalism uses it. This is getting a bit off track. Come on. Biogeo manifestation of catabolism. No, we don't want to use it in all those pathways. Syntrophy. Syntrophy is a situation in which 2 different organisms cooperate to degrade a substance that neither can degrade alone. Syntrophic. There's some lactates. So organic carbon, and then there's some lactates in here, which will then go on and produce your CO2, and some produce iron, not lactobacillus. Secondary fermentation, hydrogen, plant following figures, groundwater. That's similar to that chart 4. Rate limiting steps and time scale of chemical breakdowns. Single cell identification of microbial communities. That's how to do that. And some genes, 2 different types of cells. Yes, I won't talk about microbes. I was just going to start and just elaborate on that. But no, I did that a few shows ago. It was a big microbe session. I didn't even go through these slides. Isolation of pure cultures. Oh, enrichment cultures. Okay. Probable numbers, that's how to divide them. Alternative methods for isolation of pure cultures. Identification of microbes. Yes, it's fun to try and identify them. There's only a few, they get identified by their shapes and from there, where they are situated. See, like light ones, phototropic. So they need the light. I'm not talking about microbes, that's right. Lactobacillus. Let's see over here. Any lactose in the house? There's none. Over here, oh, Acidophytus, that's getting down there. But it's still not a lacto from the genre or species. No, it's a genre and the species are different. Come on, there's lactose. Lacto. Oh, this is enrichment culture. Oh, this is how to make it. Okay. All right. Well, there you go. If you're into brewing those things, this is how to do it. You need an electron donor and a nitrogen source. So you'll use oxygen as electron donor, and then out of here they'll use their amylase or break down the glucanase, and they'll transfer that into a source where they can harness the nitrogen out of it. Lactate and yeast extract. pH of 5. Okay, so there you go. So there's a pH that's a little bit lower. If you want to make lactic acid bacteria, your pH is 5 also for that sunshine. So this is an enrichment culture. So this is if you want to make more of them, you'll add this and at that level, and that will enrich them and they'll be stoked. So that's the areas they like to live at. So that suggests to me that if you're going to put pH 5 in the soil, well, you know, pH 5 is beneath. You want it at pH 6.5 to 7.5 is around the neutral range. There you go. That was a good little bit of a help. Went through a fair bit of slides to get that answer. Summary. Taxonomic markers. Yes, it's cool to put markers in stuff. You can stain them. You can use little. Then you need you don't need always a fluorescent microscope for the stains. You can see them under microscope too. Methyl blue. It's a very good one. Righto. Limitations of microscopy. Yes. See, while we're on it, this painting was painted from microbes. They put the smears on the Petri plate, and then they smeared it that way with all their stains and stuff in it, and look what it created. It's a microbe artwork done by microbes. It's cool, eh? GFP. All right. Come on. Any more lactobacillus stuff? No. Next step, amplification. Oh, this is for testing. This is pretty cool. This is but it was not talking about microbes. And it's a rad cartoon. I like rad cartoons. There's two words here. So bioremediation can be broken down into, this is microbes again. I won't talk about that. Transformation into microbes. Yeah, this is sweet. See this down here? What are you guys seeing? Do you see 3 planks or do you see 4? Huh? Is there 3 here or is there 4? What's going on? Whoa, that's your word against mine, mate. That's a bit of trickery here. Synthetic organic molecules. No, it's surfactants. Okay, I'm going to stop this one. I hope that's helped. Sunshine asked the question, is yogurt or fermented milk good for soil? So that's what I was showing some slides related to that. So I'll go up to here. Good. So g'day to Dave. Hello, hello. Floods and hazards. Hello, hello. Cloudscape mystical. Sunshine. Hello. Walking lightly. How are you? Hello, hello. Hello. Good morning. Morning, Kruda. Hello. Polyformus. Wow. Hello. The surface of leaves are typically the location of lactobacillus. All right. I won't touch that one. The name. Wow. Artist name. Yeah, artist name for that microbe painting. That was microbes. I don't know. There's many different types to do that. Pretty cool. Good evening to Main Green Grower. How are you going? Oh, yeah. Sunshine says, can you talk about ferments in general and their function in the soil? Oh, well, Not really. I'm not that experienced with fermentation. The Korean natural farming is a way that they do it. Main Green would know more about this, probably than me. I don't get into the ferments to put into the soil. That's getting into the anaerobic sort of style stuff, and I just don't really touch that. So it's a question I can't really elaborate on. I could show some slides and try and bring, like I just did before, but from experience on this one, I don't really have any experience of doing fermentation, Korean natural farming at all. I always done it with air pumps. I've been scared to brew up with ferment with anaerobic conditions, or when that goes into a, because fermentations, when they're changing the electron acceptor from oxygen to another source, and that's usually it's CO2, because I do fermentation with kombucha. But relationship to the soil, well, it's of an acid, and an acid's, I can talk about pH of the soil, because we know that the fermentation is of a lower pH, and that showed that in that last slide, that it was down to 5. All right, let's show some, let's go back to a pH section. Soil porosity, salinity, maybe in this one, it might be a pH section. I'll just have a bit of a squeeze before I show you. And I'll elaborate soil acidity. That'll do. I think that's pretty pH-y. What do you reckon? Yeah, I reckon it is. Oh, yeah, cool. All right, we'll just go on with that one. So if you've got an acid that you're wanting to put into the soil of a lower pH, this is what will happen. Hang on, I'm just going to share some slides. So I'm going to talk about some pH and the acidity of soil with some slides. Where are your slides? Soil acidity. The soil acidity is measured in pH, yes. It's the negative logarithm of the hydrogen proton. So this is just a general class of where it is. So you want to be the 6.5 to 7.5, and the reason for that is it unlocks the available nutrients, as we'll see down the track. And if you've got tea that's in the 5 range where the lactobacillus is, see up here, milk's about the 7, they reckon. But those lactose like to be cultured at 5, which is a strong pH, which you'd normally see in forest soils or humid regions of aridable soils. That's where you'd expect that. So that's how to read this chart. All right, next, soil sources of acidity, carbonic acid and organic acids. See, it's the acids. The accumulation of organic matter, because it's the acids, and nitrification. Do you have that nitrification process happening from NH4 from fertilizers? Sulfur oxidization also changes, and the sources may be pyrite. Actually, I'm getting into it too much. Acid rain. Plants uptake with cations, they do an exchange with the acids, because when plant roots take up more cations, they tend to balance it by releasing a proton, and that lowers the pH, because the plant roots release exudates, and in that it's organic acids, and the organic acids has the H+ ion, and that will be exchanged in the cation process, and that's where you get this exchange. So the plant will be sitting there and with its acids, with its H+, wanting some ammonium, and then that will do an exchange process through the cation exchange. Sources of acidity, human-induced acidity, yeah, like excess of fertilizers, acid rain, or potential acid sulfuric soils that's already there to start with. The balance between production and consumption of the main process that produces or consumes hydrogen ions in the soil. So that's, that's the processes, how it can happen by changing things. How aluminium causes acidity. It causes it because it's the strongest bonding strength of the cations. So if there's anything else there, it just bumps everything else out and hogs it. Pools of soil acidity. Right, that's how to get to it. I'm getting off the track here. Residual acidity, cation saturation percentage. You don't want like maths. Relationship between cation exchange capacity and types of cations. So relationship between CEC of 9 and CEC of 21. So it's got a different acidity level and a different pH level. So really the only thing that's the pH shouldn't be changing to do with the acidity with CEC though, because CEC 21 pH is higher. Over here, see, it's the same, so it's got nothing really to do with it. But it's the acid concentration that does change. 14, 33, 62, 78, it goes up. So the amount of acid in here, see, it's still got your hydrogen attached to it, still got some attached to it, it's got none and then none. This one's got more, can have more available cations held onto it. Why should we consider soil buffering capacity? Better buffered the soil, better it resists acidity from external buffers. So if you've got your humic and your organic matter high, when you put these acidic compounds or molecules in, it'll tend to sort of stay around the neutral and won't really alter much. So if you're going to use your Korean natural farming, you might want to have this in place so it doesn't lower the pH, possibly. I'm unexperienced in Korean natural farming. Buffering capacity helps us determine the amount of lime. Yeah, higher the CEC. Yeah. Effects of acidity. So this is cool. Actually, I think the next one zooms in. Yeah, look at that. So this is the acidity on. So if you're going to get your soil pH with your lactose at 5 and drop it, and it might drop it down once you administer it into your substrate, look at the bacteria. It actually tapers down. They're not real favorable in the 5 pH. They like the 5.5 and up. And then the availability of other nutrients, if you've got it in around the 5, look how skinny some of them are. CalMag, you'll probably get a CalMag deficiency. What else? That's about it. Okay. So CalMag, that's why they always say if you're running a lower pH, so you get CalMag, just add CalMag, because it's not available. Plants can't even get it. Effects of the acidity, just went through that. Mineralization, no, talking about that. That's how to affect the acidity, how to change it, why it happens. Why do you put lime onto or dolomite onto the acid soil, and why does it raise it? You don't need to know about that. Organic matter is another, yep. How to fix acidity. So you'd use your lime, or you could use organic matter, or you're growing adaptive plants. But that doesn't happen. Alkalinity? No, it did acidity. Oh, that'll do. Hope that helps, mate. That was a good question, Sunshine. Actually, I could talk about not the acidity, what's that other one? Toxicity of soil, and why you're getting into it. Because we're into this. Let's talk about sorry about that. Before I get back into it, I'll just scroll up and see if there's any questions and stuff. Prevents excess hollow stem, reasons why it happens. Walking Lightly is asked, prevent excess hollow stem, reason why it happens. I think he's asking, why do cannabis plants have hollow stems? And that's because, and the reason why it happens. I think that's, so the reason why that happens is because of the fast growth. So if you try, that means that you're probably a synthetic grower and that you're giving it too much, not too much, but a very adequate amount of its nutrients, and it's growing faster than what it should be doing. And it grows with a hollow tube and hollow bit in the middle that you can use like as a little pipe later on. They're cool. And if you grow the same cultivar outdoors with organics, you'll find that it'll grow a little bit slower and it'll develop more of a sturdier stem. That's from my experience. I hope that helps, mate. I haven't got any slides to back that up, though. Camet 432, how are you? Jezzachronic, nice to see you. Jezzachronic says, make a fermentation with worm castings and molasses. Add your own piss too. Be careful with your own piss, because that's a very high source of your ammonium, your NH4. But the problem is, if you've got with your piss, if you've been taking antibiotics, that's going to come straight out and kill your ferment. So really be careful of what you've had that is retained in your piss, like for the last 48 or 72 hours. So if you've had anything like that, be really careful, mate. But yes, in general, it's a good idea because it's ammonium. And that smell you smell off it, that's ammonia. That means that, what's the microbe's name? Not Nitrobacter. Oh, shit. Oh, no way. It's in the Haber-Bayer conversion. It's the microbe that transforms the ammonium in the ammonification. Oh, what is that lemon? I can't see it. Oh, well, next it'll come to me. But yeah, that's the microbe that does it. No, it's not. That's not. I didn't even name it. The microbe that does it changes NH4 into the volatile gas. That's what you smell from your own piss. What's Polyformus says, Lactobacillus fermentation. This species has been identified on the leaves of tomato and has been shown to have positive effects on the growth and health of the plants. Yes. Well, all right, there's something a bit to add to it underneath. Lactobacillus casei. This species is identified on the surface of grape leaves and has been identified as antifungal properties that can help protect harmful. Lactobacillus casei is from a Japanese scientist, Casey Shirota. He named that one. That was the species. Yeah, the Lactobacillus. I think Lactobacillus is a genus though, because the species is Casey. And your previous one about being on tomato leaves, yes, they've positive effects. It'd be good to see that study, but in general, microbes that are on the leaves, because they're going to make unavailable positions for the pathogenic ones to multiply. So it would be interesting to see if they've done that against to see what the outcome is, to see if it is solely just beneficial. That's another way that you can make kombucha too, is the microbes, the Saccharomyces Devices is one of them. That's a yeast that naturally occurs on green leafy vegetables. So you can just grab those and start your cultures off that way too, because that's where they'll hang out. Good question, Polly. Good, what's it called? Feedback or help. Polyformus. Good on you, mate. Keep up the good work. Just scroll down before I go into the other stuff. Sunshines, there's a question mark. Fermented tomato is beneficial? Question mark. I don't understand these bloody questions these days. Is fermented tomato is beneficial? Fermented, oh, I'm sorry, mate. Fermented tomato is beneficial. I think you're making a statement, not a question. Okay. What else down here? Cloud state. Careful. Careful, Sonny. Okay. I don't know what that one means. Jez Chronics says, oh, paper strips and a cheap ppm meter. Bob's your uncle. That's for pH. Yeah, those paper strips are pretty cool. And with pH meters, now that Jez Chronics bring it up on this way to do it, really get this. Well, I'm not telling anybody how to do it, but from my experience, I bought a lot of pH meters and they always break because you've got to maintain it this, you've got to soak it in this, you've got to calibrate it at this, all that sort of stuff. Where my accurate way is liquid. I've got this giant bottle of concentrated liquid that I tip in and it just gives me a color and it's really accurate and I don't have to calibrate it. An old mentor told me that years ago because I saw all these meters around. I said, why are you using that? And he said, he told me that. I went, oh, okay. And now I agree with him. Monty, good morning. Monty, how are you? Top of the morning to you. It's a bit of a fermentation and pH soil soil pH theme, I suppose, we've been talking about today. Monty. Jesichronic says powdered eggshells fix that. Okay. That's hot. It's a slow release calcium in powdered eggshells. Oh, mate, where was I up to? Oh, geez. Is Kemet 432 says, is sea sand a positive or negative ion? Sea sand? Sand is a texture. It's not a chemical. So it's sand falls under the sand, silt and clay regime that you have making up your soil. And it depends on how much you've got of each other's to which one you have. So sea sand actually is just a texture, mate. It's not, hasn't got a charge. If it has got a charge to it, because of the conditions that it's under. If you dry it out, it might have like a staticky charge to it. But in general, it's not classed as an anion or a cation. Good question, Cam. Been listening from the start, says Monty. If you think, I can't come down to. That's a bit strange. Jesichronic. I don't think that at all, mate. I think it's alrighty. A bit odd. Monty's, yes, sunshine. Kemet, face, okay. Jezza, only. I'm not going to get into it, but only fools take. Some people are really sick or they have an autoimmune problem, so they might be inclined to take the antibiotics. But in general, yeah, it's good to try not to take it to build up your immune system. And those folk have good piss. Yeah, got to watch acid levels. I agree, Jez. I'm looking into ferments, says Sunshine. Sauerkraut, kefir. Going to boost. Yes, okay, to boost your own personal microbiome. Good work. I was going to say kefir for you. There's another cool one you can use too if you're getting into this health. I sort of off the medical cannabis topic. Anyway, I'm into human health too. You culture up. I did it in Canada. The Lactobacillus casei shirata strain. You put it into milk. So, for instance, you'll get one of those little tiny bottles at the supermarket. Little 2 ounce to 50 ml things, whatever they are. Tip it in, and then you'll leave your milk out for 16 hours in about 30 Celsius. You know, what's that? 90 Fahrenheit. And it'll sit for 16 hours, and then it'll do its bit, and then you'll put it back in the fridge, and then that's it. It's inoculated the rest of your milk. And then once you get down to your, say it's a 2 litre or half gallon jug, you'll get down to the bottom of it. You'll save 1 or 2 cups, and you'll tip that into the next milk and do the same thing. You can continue your culture along and do it that way, mate. That's another cool technique. But it's very beneficial because, yeah, your human gut microbiome is so important, and the better you can work on it, the better you'll be looked after. That's another topic of my study and interest as well. But this is medical cannabis, so we'll get back to it. Walking lightly. These are growing great. Just throwing out some pondering points. Good work, mate. Well diluted after sitting a week. Okay. I got into it a week ago. Many spices available, and you can multiply them. Yes. 1 capsule in with that is good. Okay. Very good, mate. Glad you're having some luck, Jesichronic. Oh, here we go. GMF. Nice to see you, Genetic Memory Farms. This person knows about, he works with lactobacillus cultures, so if anybody is going to know, this person will. So the common lactose strain that is on the surface of most vegetables is Plantarum, Lactobacillus plantarum. It makes sauerkraut, kimchi, pickles, etc. All right, and that's Kemet's got a little his recipe for his recycling of his urine. Very good work, mate. 1 part is to 20 into your reverse osmosis water. Very good, Kemet. Nice work. Sunshine, yes. Good evening, peeps. Oh, there we are. You can make perpetual fermentation too. A kombucha system is for that. Yeah, I do kombucha. It's rad. A lot of people on this in the stream too do kombucha as well, Jazzachronic. It's for good health, mate. Dave to you. Hello, Dave to you. Titration. He's into his titration. That's his statement. Very good. Lots of good info. On to, okay. Well, Sunshine, at least you can watch the show back. Hey, mate. Woohoo. Here we go. What's Germanic Mary Farm say? Perpetual pickles are commonly the Chinese style. Just add clean veggies to the same brine for years and years. Jazzachronic says big glass decanter 15 to 20 is also good. He's researched, or they've researched that, and they like it bigly. I deleted that message. Janae, yeah, perpetual tea is easy. Cheers, cheers, and cheers. Cheers up, the simplest and better. Rock dust or rock sand balance pH, so I can believe it's negative charge for a plant. What's that about? Kemet says sand typo. Oh, so rock dust or rock sand pH balance. So I believe it's negative. Oh, okay, rock dust. Rock dust, yeah, that's where you want to get your phosphorus out of that, and you want your chemolithotrophs. They're the microbes that will be your phosphate solubilizing bacteria. And you'll have those ones, and then they will break it down, and the chemolithotrophs, depends which ones you have and to what oxygen, what pathway they take to the outcome of the pH. I think it would be all right in general. So what's the question? Rock dust or rock sand pH or balance it? Oh, so they buffer it. I don't know about that one because it takes so long for the rocks to be broken down itself. It's, what do they say? It's 1 million years of rocks to be broken down to get 1 gram of soil, something along those lines. So it takes just after its weathering process and it's goes through its transformation process, it takes a long time. So I'm not sure about that one, mate. So I believe it's a negative charge for the plant. Yeah, I think that it's such a minimal effect. Oh, but if you put rock dust on it, well, if you mix the rock dust with, say, water, that would be forming, and depends what type of rock dust. Because if you put your, what is it, your pyrite dust on it, it'll break down and form a amylase, which is simulates a starch breaking enzyme. Anyway, I think I've gone into it a bit too much. Rocks, dust, so I believe it's a negative charge for the plant. It would be minimal, I think, at best, mate. That's my personal only opinion. Thanks for input though, Kemet. Hello, Gobby Cannabis Vaporist. Good morning to you. Thank you. Hello, Jeff Papalia. How's it going? It's been late scanning the girls for mold. It's been late scanning the girls for mold in this crap weather. That's no good, mate. You need some mold resistant or fungus resistant genetics, my friend, to put into your genetic pool. It works very well. I've got some videos on it. Oh yeah, Polyformus says silicon oxide is sand. Yes, it is. Yes. No frills, rock dust, awesome product. That's the way, Jezza. Nice work. Rock dust. Oh, okay, that's why you're asking for it. Ah, yeah. Well, you can do a test with that, mate. Do your own test to see what sort of specimen you've got. You put the rock dust in, you pH the water first and make sure it's, well, whatever it's going to be, probably be nearly 7 or 6.5 or whatever, 7.5, depends where you are. And then you'll put the rock dust in and then let it settle. And then pH it at, say, 1 hour, 3 hours, 5 hours, and keep writing these numbers down at that level. And then you can see for yourself how it alters the pH. And then please let me know what the results are next week or whenever you can, because that'd be cool just to know, because I don't know. And I think remember what I said, my hypothesis was it would, if it's going to alter it, it would alter it very minimally. So make sure you do the weights with the percents on if you put in, say, 1 gram to 100 mils of water or something like that, so we can work out what percents it is too. Nice work. There's a scientist in the making, Jezza Chronic. Walking Lightly says, pumpkin and comfrey ferment, anyone giving it a go? Wow, pumpkin, is that for the plants or for you? It sounds like he's getting into it himself, but I reckon he's talking about plants. Oh, yeah, 100 and, all right. Morning, Jeff. Yes. Jeff says, yes, I do. CC mold resistant plants would be awesome. Yes, you can, if you're into breeding. We're going to have a chat sometime, Jeff. Let's just have a chat. G'day, Nick Kelly. All right, back to it. Now, I was going to share the screen after all that. I don't really remember why. Oh, that's right. It was to do with toxicities in soils. There was one of these ones. Now, if I go right to the end, actually, I think it was the other one. There it is, pesticides. Oh, it's not really. Synthetic organic compounds in soils. So by putting strange things into soils, what happens? So xenobiotics is synthetic organic chemicals which are unfamiliar to the living world, because xenos, the Greek word for strange. So it's strange living things is the breakdown of the xenobiotics. Differences commonly with putting in halogen items, chlorine, bromine, other equivalent non-metals such as sulfur and nitrogen into the cycle. Representative compounds in 18 classes of widespread organic compounds. So these are the, they're the industrial, they're man-made ones. Insecticides, yes, well, the flow-on effect of all these insecticides, fungicides, herbicides is bad. They don't break down fast. And that's the biggest problems. They get into everything. Their long-term effects is so detrimental to everything. Contamination and persistent organic pollutants, also called organic organochlorines. That was an exam question. An organic compound, yeah. This is a cool chart where it shows you how it actually, I feel sorry for you people in North America. Please filter your groundwater, because I think it's 90% of the lower 48 is polluted from insecticides and fungicides and pesticides from farmers put in. This is how it works. So it'll get absorbed, it'll sort through the can travel, and its pathway of its half-life is to be broken down is so high. So it'll take years and years to really break down, and some of them require particular microbe interactions to get them, like plastics requires, there's only 2 or 3 that microbes they found to break down plastics that can eat it. So it's a similar sort of thing. To break down these chemicals, they need to be in the right conditions with the right sort of microbes. It's hard to find. Absorption in the soil, this is just how it happens. So see up here, charcoal, it nearly absorbs all of it. So if you've got any problematic areas where you think, shit, something's gone down, you don't want it, grab some charcoal out, and that should house it all up. Then you can get rid of that charcoal. It's just a very fast way. Like it's safe with humans. If you ingest something, some drugs or anything like that you don't want, a quick fixed method is charcoal tablets, and that absorbs anything that, and it holds it in the charcoal. Yeah, soil organic matter is another really good one. Montmorillonite is the type of clay with the 2:1 ratio, which can absorb it and hold it, lock it in the nutrients as well. Land salt and hydrogen peroxide treated salt can work with it. The soil lost most of its absorption capacity when treated with hydrogen peroxide to remove its organic matter. So those people that want to put hydrogen peroxide down as treatments, there was someone a few weeks ago that said, oh, we should use hydrogen peroxide to kill the microbes in the soil. Well, it's true, you will kill the microbes, that microbe, but you're going to kill everything else too. Pesticide effects on soil microbes. The effects on microbes, well, it's just high. Here, the burrowing Effects on soil, on organic pesticides on soil organisms may be more subtle than mortality. So here, the burrowing behavior of earthworms was shown to be dramatically inhibited by the neon inocadol, by an insecticide often used for seed treatments. So you see on the left here, the control, living, beautiful. Here's 0.1 milligrams per kilo, not much. And in here, 0.5, look at that, it's just virtually non-existent. And there's, of course, there's a human effect. And cannabis being a biodegrader, it's a bioaccumulator, it'll absorb this if given the chance. So then we'll ingest it. If you're going to extract, you'll can be taking some of those actually into the body. Be careful. All right, that'll do for that bit. Actually, there's another section too I could show you on the, I'll see what these reckon. Jeff, all right, hope that helps. Plant resistant. What's the genetic memory farms? The mineral uptake process goes faster if you run air through water used to make your own limestone water. If no air, the ppm will climb much slower. Okay, the mineral uptake process goes faster if you run air through water used to make your own limestone water. Okay, he's making his own limestone water with air, I think. Okay, that's good, mate. Jezza says stinging nettle, FPJ. So stinging nettle fermentation I made once as a fertilizer. Yeah, nettles actually it's quite good. I used to eat nettle. It's got more iron, 30 more times iron than broccoli. It's a good substrate, like comfrey too. It's good compost additive. I do pumpkin compost. Oh, here you go, walking lightly. Kemet says he does, or she does, pumpkin compost, pumpkins for the neighbours. There's a bit of interaction for you. Sucks bad, toxic, everything. Yes, it does. Kemet says fungi wash many OTC mushrooms, oyster mushrooms, will help you. Got to look them up though. Fungi wash many, I don't know what that means. Aussie photos. CC, why did you... Right. This shows into... it's not a drama show, mate. We don't discuss what's going on. This is a medical cannabis show, mate. GMF. Just using Airstone. Takes me a week to get 20 ppm water up to 120 ppm with powerful limestone. Oh, there you go. So if you want to make your limestone water, that's what GMF uses with success. Aussie, you filth. Oh, wow. You just learned how to get a block, didn't you? I just said no drama on the show. And Jezza Chronic. High silica in nettles is what the little tiny spikes are made of. High silica. Oh, wow. Wow, that's cool. So that's broken down. You get a little bit of bonus silica. That's nice. You don't have to use your sand. Off to dinner. All right. See you walking lightly. I was going to show... Oh, that's right, the other pollutant stuff. I'll try and bring that up. Where is that? Just to finish it off. So that was in, I think it was this one. I hope it was right at the end. There was a big pollutant section. It wasn't on much, but it was, yes, cool. Absorption of pesticides in soil. Oh yeah, I suppose it just adds to the pesticide sort of thing. Let's see what's in these few slides. Share screen. Absorption of pesticides. Here we go. this is just the follow-up on the pesticides one, I suppose, related to the soil. Soil can absorb pesticides through the ion exchange, through the anion exchange capacity or the cation exchange. Absorption can reduce this into the groundwater. It can allow time for soil microbes to break down chemicals to less toxic byproducts. It is common for organic compounds to be absorbed within soil organic colloids, so the actual soil particle, by a process termed petitioning. The hydrophobic, meaning the water, hydrophobic part will not be absorbed by most clays, meaning that it rejects it. It can't be absorbed because it's hydrophobic. That means it's, well, phobia means, you know, you've got something that you don't like, and then hydro is water. So it's water that you don't, something that you don't like. Petitioning, not going to go into that. Organoclays. The hydrogen metal cation, calcium for instance, that are absorbed on the surface of smegmites, clays, can be replaced with large organic cations given to the road organoclays. Okay. So this is a picture of a field in Africa, and after applying the herbicides, the whole field was still full of weeds. And why? And it's because the soil has, in the upper A horizon, it's got a really low, so in the first, in the upper A, O horizon it goes, organic, and then it goes into the A horizon. So that first sort of 0 to 15, 20 centimeters for the first, you know, half a foot, the soil has a very low cation exchange and a clay subsoil, which is a high cation exchange. So that means all the pesticides was absorbed by the subclay and not the top surface. So therefore the weeds of the roots, they go down only just in the top surface, and so they had no chance in getting an effect. So this is an example of where they sprayed it and all they did was just pollute the water table below, and they had no effect. So that farmer probably would have went, oh, I'll just put more on, you know, I'll just try it more and more. So it just pollutes more and more, which is so sad. Where if they just would have got out and put a decent ground cover or got something other to combat it with, they'd have a better microbe interaction with their soil. Distribution, this doesn't go for very long. KD and KS, okay, several. There's the different effects of the binding and biomolecules to clay and humus. Bacillus thuringiensis, that's a cool microbe that can absorb the toxin. And BT, it's another, that's really good for insects as well. So it's a good little addition you could have to your outdoor crop if you're in this type of situation. So if you went to a place and it's non-organic and you want to try and make it organic, you might think about putting Bacillus thuringiensis into your IPM program. Scanning electron microscopes of DNA from Bacillus subtilis bound to colonelite clay, which is a 1:1 ratio on the left, and then to montmorillonite clay or smegmite clay, which is on the right, which is a 2:1 ratio. The arrow points to the strands of bound DNA, so you can see the arrows there, and these ones in here. And the DNA bound to clay or humus is protected from decomposition by retaining the capacity of transferring genetic information to living cells. So that's Bacillus subtilis has just gone and done that. So it's very beneficial. Antibiotics are also absorbed by soil via the CEC process. After absorption, after it absorbs in, see, you know, there's adsorbed, which is on the surface, and AB absorbed, B for Bob, and that's into the surface. So after it's absorbed, it develops positively charged sites around in the soil. Increasing research shows that even through absorption to colloids may reduce their efficacy somewhat, the soil-bound antibiotics still work against bacteria. There is evidence that at least some antibiotics can be taken up from soil by food crops and so enter the human food supply. Not good, let alone killing all for your microbes, which you're trying to develop a soil food web. It will also increase the resistance against life-saving drugs in the body. Yes. All right, I'm not going to get into the actual different types of ones. So that's the last slide. Sweet. So that was a good little rundown of soil pesticides, I suppose. Is there any little additions down here? Use fungi on toxic runoff. That's all. Okay. Yeah. Okay. That's what Kemet says. Exactly on the soil side. Okay. They could run a wheat crop in Africa and drop it into the soil. Oh yeah, from the Africa slide. Yeah. Yes. All right. Well, I don't think I need to show any more stuff. I'll just wait a bit. Do you have any more questions on, I suppose, pesticides in the pesticides path in the soil? What else was there today? Fermentation. A bit about microbes again. Microbes are so cool. So I'll just give it a little tiny bit because there's a 30-second delay that comes through here. And then we can see what's going on. Let's see if I can possibly wrap it up or show some more slides. And next week's show will be a open topic again. So if you've got any other things that you'd like explained or we can discuss on medical cannabis, please save them till next weekend. And this would be the same time next week. And that little money button underneath is for appreciation, and I thank you to the people that have done that and clicked it. It's good. And the subscriptions are there available too, a few different categories that comes up when you click it. Thank you for those who have done that too. It's good. And yeah, next week I'll be doing the same thing again on open medical cannabis topics somehow. I'm just showing all the slides. My uni stuff's nearly wrapping up this semester, so it's good. I'll have a bit more time to put in to making these shows a bit more news related, I suppose, like they used to be. Hello. G'day, Ricky. Wow. How are you going, mate? Anything? Kemet says anything on electrification. Well, electrification is to do with volts or millivolts, you call it microvolts nearly, and that's to do with the redox tower and the cation exchange and anion exchange and how they develop their charge. So that's a full-on topic in itself. Maybe we can do that next week. I can talk about the charges in soil and the cation and anion exchange on their bonding strengths, on which ones do it, which ones knock each other off, why sodium is at the bottom of the chart, and why aluminium is at the top of the chart. I can do something like that. Oh, wow, look at this. Hey, Google E. Thank you, Google E. That's appreciated. Good on you, mate. You have to write down a topic for the show that you want discussed or something. I haven't spoken to you in ages. Thank you, Google E. Very nice and it's appreciated. Very good. That's really good. Good on you, buddy. Google E. Dave says, Google E, the man himself. Thank you. Oh, yeah. Dave says, oh, sorry, Kemet says, I need to know more of that. Okay, so that was about the electrification and the ion exchange. Okay. All right, well, how about I title next week's one, that? Unless Google E's got something to say, hey? But I reckon I'll put on we can go through the ion exchange and the soil voltages. And I've got a fair few slides on them and talk about why it happens and how it happens and what's the best voltage to still to stay in. And can you discharge the voltage? Can you put out earth stakes and try and, discharge it and get things done a different way? There's a few different cool things with the voltages that you want to do. You want to always, yeah, anyway, that's a good idea. Good suggestion. to Kemet. So yes, I'll title that, and I'll try and prepare some slides for next week so we can have the, actually I'll better write that down because knowing me, I'm not the best. So it's going to be your iron IEC and your soil voltages, soil V, no worries. There you go, it's written down so I won't forget now. To Kemet, and yeah, thanks. All right, there was no real questions coming through. He just says he needs to know more of that. Have an awesome day, everyone, says Lene. Yes, I might do it too, and just say to thank you. I appreciate everybody rocking up and putting in the effort, and please join me for this time in 7 days' time, or in not 167 hours, because it's 168 for a week. So yeah, I'll see you in, I hope to see you in 167 hours. I appreciate everybody. And Googly, thank you for your donation, mate. That's really appreciated. And we'll see for next week's ion exchange and soil voltages. We'll talk about that. Yes. The American one. Oh, you just missed out, mate. Just leaving. So yeah, check out electroculture for next week too, please. It says American one. Yes, but that's what I'm going to try and show some slides on that and just go through some things that I have on that. So we'll discuss it. And yeah, thanks again, everybody, putting in the effort. I will catch you all next week, I think. Thanks, Jeff says, see ya. Renee, see ya. Thanks, everybody, again, for rocking up. It was good. Googly, thanks for your contribution. It's appreciated. And we can see next week's title to show, ion exchange and soil voltages. And of course, there'll be more questions that you guys can and girls can ask if you want. Everybody's welcome. No drama here. We just talk about medical cannabis and how to bring out its genetic potential and in a safe and healthy way, so we can get the best out of the plant and for our bodies. Thanks, everybody. Peace out, says Jezza. Excellent. Peace. Very good. And peace to all you guys too. So happy breeding, happy growing, and good day to you all. Bye-bye.